Hello everyone, my name is Guillaume Salzet. I'm doing my PhD at Beta Economics Lab in France and also at EcoFog Research Center in French Guiana. For the 58th ATBC session, I will present you a part of my PhD work on how hydro-edaphic gradient shape community of trees in a neotropical forest of French Guiana. To give you a brief context, tropical tree communities can be viewed as the result of deterministic and stochastic factors. Deterministic processes are niche-related and tend to distribute species in their optimal habitat. That's called the habitat filtering effect, represented in this figure. In the case of French Guiana forest, located in the Guiana Shield, many studies have supported the importance of habitat filtering via topographic variation in tropical forest, as it promotes niche differences. Topography creates differences in hydro-edaphic environment, and as water availability in soils is a strong driver of species distribution, represented here with drops, this hydrological environment were found to modulate, for instance, the drought stress. On one hand, we have hilltop with terra firme forest, where water availability is constrained in dry season. On the other hand, we have bottomland with seasonally flooded forest, where water table is close to the surface, but can cause hypoxia stress in rainy season. Based on topographic gradient, it does exist discrete species habitat preference classification. It classified species into habitat specialist, bottomland specialist, hilltop specialist, and generalist if it doesn't show clear habitat preference. From this classification, it's difficult to estimate what would be the composition of an intermediate topography community, like here. That's why we should move to use continuous hydrological grounded indexes. The advantage of continuous measure of a gradient is that enable to follow community change, especially in intermediate location. So we need a continuous proxy to describe the hydro-edaphic gradient. In literature, there is a previous study of Kopersky, Masek, and Wild in 2021 that promote to use Topographic Wetness Index, which quantify a steady state of soil wetness based on runoff water fluxes from hilltop to bottomland. This index has been used in French Guiana to describe how a set of species complex can be spatially distributed. To give you the example of Symphonia complex, we have two very close species, genetically speaking, Symphonia globulifera and SP1, which exhibit a strong niche differentiation along the TWI gradient. On the hilltop, almost every Symphonia tree belongs to SP1, but if we move gradually to bottomland, relative frequencies of SP1 and globulifera are gradually reversing until bottomlands, where globulifera is found exclusively. We can wonder if using TWI could help us to detect other cases of hydro-edaphic niche differentiation. That brings us to the following question. Can we describe tree community composition as a function of TWI? If it works, is the TWI consistent with species habitat preference classification from literature? If it's consistent, can we test if TWI effect is conservative along the phylogeny? And fourth, could we test to detect other genera that show a similar niche differentiation mechanism, just as previously shown? To address this question, we relied on spatial inventory from Parakou experimental site in French Guiana. We used the 2016 census. We retained species with at least 10 individuals over the experimental site. We used almost 35,000 trees botanically identified. We also used habitat classification map from Ferry et al. In 2010. We computed a topographic wetness index map from high-resolution digital elevation model derived from airborne LiDAR campaign. Using slope, flow width, total catchment area SAGA module, we computed a TWI map, which is consistent with the habitat classification map. Taking advantage of spatial inventory, we designed a joint species distribution model at individual level. Each tree is botanically defined and TWI is associated. In this model, we assume that the observed species is a draw for a categorical, and this is linked to a species probability vector, which is computed using a softmax function. And this is linked to the from a relative species frequency effect and also a TW, an individual TW effect, which is computed for each species. We can interpret the TWI effect as a habitat preference. If it's negative, the species would be more frequent on hilltop than bottomland, and if it's positive, it would be the opposite. After the inference, we will found that the site R square is about 87%, whereas we have plots R square and subplot S square that show little drops from this level. So we can validate our first hypothesis. Now we want to check the consistency between TWI effect and species habitat preferences classification. We selected the up-to-date classification from Baradotto et al. In 2021 here, and each habitat preference are measured with an association level index called unveiled here. And we have here the 5 highest unveiled for bottomland specialist and hilltop specialist. When we checked, we found a positive correlation between TWI effect and bottomland association index, but we could not find any correlation with hilltop association. So for now, we found that TWI gradient provide a good overview of local assembly, local species assembly, and especially for bottomland specialists. For the following part, we will focus to measure phylogenetic species signal between TWI and phylogeny. We use the openly available plant megatree from Jean et al. From 2019, and we computed a phylogenetic choreogram to detect if TWI effect would be aggregated at a given phylogenetic distance or dispersed. But we know that at short phylogenetic distance, we have a nosy estimate of this correlation. That's why we had a variance partitioning to check if we missed a large part of the variance at species level. And we observed that the phylogenetic choreogram is significant at short distance, but we found in the same time that species concentrate the main part of the variance. So we have to investigate the intragenous specialization to understand what happened at the species level. To test, we could detect if we could detect the same observed pattern like the species complex, we rescaled the TWI effect at genus level and look if we could detect or not a differentiated pattern like this. We found the same result as previous studies on species complex with a clear differentiated pattern, significantly different. And we found other genera that found a nosy differentiated pattern. At least a numerous of these genera have a one significant bottomland specialist. We found another set of genera where we can find only one specialist at 50% significance. We can wonder if it's an artifact or not, or a real specialization of only one species. In the end, we found many species without any specialization, but we can observe that many genera with more than 2 species seem to show at least one specialist. To discuss about the limits and enhancement of this model, we can begin with H1. We know that it does exist special pattern with aggregated species that should be tested, and that's the limit of this model. We found mismatch and between TWI effect and habitat preference classification. We need to check with other classification at broader scale to see if this mismatch is repeated. Phylogenetic signals have been found, but we need a high resolution phylogenetic tree to be sure and at short distance. Niche differentiation along TWI have been found in older genera than previous studied, but we need to add other strategic gradient to improve prediction, especially in hilltop. Finally, it seems that TWI is an adapted proxy to describe neotropical hydro-edaphic gradient. We validate H1, H3, H4, and partially H2. TWI could be used more frequently because it's solely based on topography and exhibits a strong impact in species assembly. It allows us to describe habitat preferences through phylogenetic view and niche structuring mechanism. Thank you.